Hello, my name is Ashley Zhou, and I worked in the Gardner Chen lab under Dr. Kellen Chen, where we primarily investigated how diabetes affects wound healing. So, over 200 million of the 830 million people with diabetes have impaired wound healing. This means that diabetic cells can't respond to insulin, which leads to high blood sugar levels and a weakened immune system, which weakens wound healing in general. However, some individuals are able to heal effectively, and we wanted to see why. Our past research on mice showed that there was a high expression of these immune-related proteins CD14 and CD64 in the non-diabetic tissue and a low expression of those in the diabetic tissue. Therefore, my lab specifically applied this to humans to see if the trend was similar. To do this, we first collected tissue samples from non-diabetic, which is a control, the diabetic healing, and the diabetic non-healing from consented patients. After that, we froze the samples and then we sectioned them by using a cryostat and then stained them, and then we imaged them using a confocal microscope that allowed high resolution for us to see the proteins clearly. Then we quantified them by using a MATLAB code developed by our lab. And the results show that there was actually a higher expression of CD14 and CD64 in the control tissue, and the difference was really significant in contrast to the diabetic groups. However, something unexpected we found was that while there was a higher expression of CD14 and CD64 in the diabetic healing in contrast to the non-healing, the difference was not significant. For future reference, we plan on seeing how these proteins interact in the wound microenvironment so that we can use them for future treatment. Wound healing is a natural process that is impaired in diabetic tissue. However, through our research, we're closer to finding the right treatment so that these individuals can have the same opportunities of healing fully. Thank you.